Okay, this project has to do with normalization of gene counts. And I say hot off the presses because this is a problem from ongoing research. We're working on a certain project and this issue has come up. If you want some background to the method, you can look at this article. And I'm not going to go into too much detail into the biology, but just you know that cells have genes, and when cells reproduce, the genes reproduce. And each gene expresses several times. So you have the gene being instantiated multiple times. And the number of times that a gene expresses has biological function. Now, gene expression level data can be used as markers for cancer or other types of diseases. Cancer causes uncontrolled reproduction, so sometimes certain genes are expressed more than usual or less than usual because of cancer. So diseases may enhance or suppress the expression levels of various genes individually or in combination. Right? So gene expression level data, what that is you have a list of genes and you have a number, which is the number of times that is expressed when the chromosome is reproduced. I can show you some gene expression level data here to give you an idea what's going on. And I have it here. This may take a minute to open up. It's a rather large file. And here what we have are the gene symbols listed on the left. And these are sample identifiers. Typically, you're taking samples from cells from different patients. And each gene has an expression level. And you can see that different genes have very different expression levels. Look at this gene here. You can see the expression of this gene is quite low. In contrast, this gene tends to express very high across patients. And it's not completely consistent, but you can see that there's a relative level. And then this one is somewhere in the middle. Okay. So different genes have different characteristic expression levels. And we want to see, to find some way if we can identify when one person's gene expression level is much higher than normal. For example, if you look at this gene here, this 30,000, here I have 30,000, and here we have 3,000. Now, does that mean this is abnormally high? Well, if you look at these other instantiations, it's very variable. So it's not really clear if this is abnormal or not. You would have to do statistics on this gene. Right? So that's what we want to do, is compare gene expression levels across patients. Again, the patients are in the columns, the genes are in the rows. Okay? So that's the kind of data we're looking at. Okay? Now, in order to understand how this is working, we need to understand a little bit about the process of these measurements. So genetic expression levels are measured by a very complicated process. So basically, and this is very oversimplified, a sample is taken from cells in a patient's tissue, which consists of a pool of gene fragments. Okay, so you take a sample. Let me see if I can draw this. Okay, I should be able to do this, sorry. Sorry. Getting used to this. Okay. Here we go. So let me just draw. So here's my sample. And then inside that sample, I'm going to have different gene fragments. So suppose we have a certain number of red fragments, and these fragments are all from a particular gene. Then these fragments might be from another gene. Okay. And then these fragments might be from another gene. Okay. All right. So these fragments are from the gene. They may be from different instances of the same gene. All right. But so what you want to do is you want to count the fragments, and then you want to map the fragments to the particular gene, and that gives you a gene count. There's a little more to it than that, but that's kind of how it works. Right. So you have a pool of gene fragments. So sorry, let me get back here. Here we go. Okay. So a large number of these fragments are sampled randomly and mapped to the corresponding genes. So that's my picture here. So fragments mapped to genes. As a result, each gene is assigned a certain number of reads. All right. So that's the read data that you saw in the spreadsheet. So to compare gene expression levels between patients, for each patient's sample, the gene reads are divided by the total number of reads to get the proportion of total reads corresponding to each gene. So I made a little toy example here to show what I'm talking about. Right. So here I've reorganized the data a little bit. Here I have the patients in rows and the gene expression levels in columns. So that's different from this other database where I have the genes in rows and the patient IDs in the columns. Right. So what I want to do is I want to take the gene reads for each patient and sum up, and then to find the proportion, I will divide the value by the sum. So here, if I take these here and I take the total reads per patient, this would be the sum of these reads here. And let's make this bigger so you can see what's going on. Okay. Here we go. Okay. And then see, you can see the formula there. And I'm going to fill that formula down. So I'm taking this sum for all these different patients. And if I want proportions, which I can put down here, sorry, this would be gene 0, gene 1, and gene 2. I would take the value here, take the value here and divide by the proportion, which is here. Now, if I want this formula to copy, I want to make sure it's always in column E, so I'll do this. So if I do this and I fill over, and then I also fill down. Okay. So I just copied that same formula into each cell. You can see it's the same formula. If I take the total proportion to check whether I've done this correctly, right. Now, you see that the total reads per patient are different from patient to patient. So really, I can't just compare these numbers directly because for instance, I might have a relatively, this 27 out of a total of 400 or 500 may be less than this 12 out of a smaller number, right? So we're looking at proportions here. Right. So in order to compare across patients, I want to take proportions. Right? But there's still an issue with proportions. And let me illustrate that by jacking these numbers a little bit. Suppose this number here were very large. So if I look down here, this proportion has been updated. The proportion of gene 0 for patient F is quite low, even though this number is rather high. In fact, the proportion here is lower than the proportion, well, it's comparable to the proportion for A, even though this is more than 3 times as high as that. So the problem is that this exceptionally high read for gene 1 has messed up the read for gene 0. So just taking proportions causes a problem when there are genes that are highly variable. As I said, the high gene read on one gene may mess up the proportions for the other. And that's what I mean down here, is that some genes normally have wildly variable expression levels, which produce large variations in gene reads for these genes. So if one of these wild genes has an exceptionally high read count, it will reduce the proportions of the other genes. So the question is, can we improve the normalization to mitigate this effect? Do we really want to just divide by the sum, or is there a better way to do it? So before trying to figure out how to improve the normalization, we should think about how to test this method and see how well it works. So the way to do this is simulate. All right, so here's where modeling comes in. Now, in olden times, people would try to figure out the underlying probability distributions to use in a simulation. So in order to generate this data, we'd say, well, I think patient genetic data follows a normal distribution or log normal or exponential or something like that. However, in modern times, we can be a lot dumber because thanks to the computer and massive amounts of data, we'll just use our data to generate the distributions for us. Right? So the idea of this is that suppose you do have data that's normally distributed. If you have a lot of data and you have normally distributed data and you take a histogram, the histogram will look normal. So then rather than trying to model the data as normal, I will simply use my histogram to give me the distribution. Right? So in fact, if my distribution is not normal, then the empirical data will give me a distribution that's representative. So in order to do this, you do need computer and massive amounts of data. So we're just going to let our data generate the distributions for us. These are called empirical distributions. And the method for using empirical distributions to create simulations is called resampling. And so we're going to simulate by resampling from these empirical distributions. All right, for our empirical distribution, we will use a database of n patient samples, where each sample has n genes. All right, so that's our big database here. And I will make this available to you. So the number of genes, if I want to find the number of genes, I'll go down here. So there's over 20,000 genes. The number of patients is not as easy to count, but it goes over here. I believe it's 408, something like that. It goes up to column OT, whatever that is, but that's quite a bit of patients, around 400. Right. So, all right, so let's see. So you can read this information into a NPAT by NG NumPy array, which we will call read matrix. Right. So notice that the original database, as I said before, has genes in rows and patients in columns. So you will have to take the transpose. So that's what this says here. Okay. All right, now I won't tell you why right now, but after reading in this matrix data, I want you to resort the columns so the columns are in order of increasing standard deviation. If you don't know how to do that, you can use ChatGPT to help you with that. So what do I mean by this? Well, just to show an example how that would work here, I would take the standard deviation. And in Excel, they don't have separate standard deviation for population and for sample. So let me finish this standard deviation. And I'm going to take the standard deviation of this column. All right, let's see if that works. Okay, let's see if that works. B3 through B9, okay. All right, so then we take this standard deviation and apply standard deviation to the entire 3 columns of my database. Right. Now, you can see that the standard deviation is not in order. Right. So what you'll want to do is you'll resort the columns so that this column, the data in this column is over here and the data in this column is over here. So I won't do it now, but that's what you would have to do. You'd have to sort this in increasing order. Well, let's see if I can make this work. Let me take this out here for a second, put it here, and then I'll take this data here, and then copy, and then paste the values here. And then here I'm going to move these values over here. Sorry, not sure what it's doing. Let's try that again. All right, so it screwed up, so I'm going to do this. All right, let's try it again. Excel does some funny things sometimes. We'll get there. All right, so here we go. Copy this, paste it here. Now paste the values. Okay, and then everything is fine. So let me just get rid of this because I don't need it. All right, so I've resorted this. Now, you would also resort the names here. You really don't need to. You don't need to change this around. I still keep this as 0, 1, and 2 because we can keep track of the gene names later. All right, so these gene expression levels should be sorted in order of increasing standard deviation. All right. And I think this fixed it up. Should have fixed it up the same way. So I think we're good. All right. So increase the standard deviation. So we can use this data to construct simulated patient samples, and that's test our idea. Here's an outline of the procedure. First, create simulated patients. Write a function that takes as inputs the data matrix and an integer nsympat, and outputs an nsympat by n gene numpy array that contains count data for the simulated patients. And we'll call that sympat matrix. So, create each simulated patient by randomly selecting with replacement a gene count for each gene from among the npat patient read counts for that particular gene. Do this independently for each gene. You should vectorize your code to make it faster. Loop through the genes and for each gene, do all patients at once. All right, so let me show you what the operation is here. Here's where we have our simulated patients. So this is, I'll call this A prime because this is not my actual patient A, so this would be B prime, my simulated patient B. All right, so in order to get A prime's count for gene read 0, I would randomly choose one of these. Maybe I'll say 81. And for B prime also, I would randomly choose another one. And I randomly choose with replacement. So it's possible I could choose the 81 again. But let's say in this case I choose the 12. So these are completely random. They're chosen from this column. Now, in order to speed up your code, you should choose these together as a single vector statement. Otherwise, your code's going to be very slow. So you're going to do the same thing here. You're going to choose one of these randomly. Maybe this one is 178. And maybe this one is 93. And for column 2, well, maybe they both happen to be 4455. Okay. Perhaps they both chose to be that. Okay. All right, so these are simulated patients, and I'm accepting this as the ground truth. I say that this patient A prime actually has this expression level for gene 0. So what I'm going to do is create observations that will try to recover these actual expression levels. Right? So that's where we're going next. Okay, where is my, there we go. All right. So that's what you do for each patient. Right? Okay. All right, so we will use the sympat matrix array generator 1 as ground truth. Next, we will simulate the process of sampling fragments from these patients. But how many fragments should we sample? Okay, empirical distributions to the rescue. Right? We have some empirical data, and we know from that empirical data what typical sample sizes are. So we can obtain npat sample sizes from our empirical data by summing over the rows of read matrix, rounded to the nearest integer. Right? So that's these total reads per patient here. All right, so I'm going to use these numbers to generate the read counts for my observations of the patients. So that's in this table down here. Right? So for my patient, I should call this patient A prime, and this is patient B prime. So my read count goes here. So my read count for patient A is randomly chosen from here. Maybe it's 5003. Okay, maybe I chose it from this number here. All right. So that's 5,003. And maybe for patient B, I chose another one. Maybe it's this one, 4549. So these are going to be the read counts. Again, these are the counts. This will be my simulated observed data that I want to infer my simulated patient data, which would be my, as I said, my ground truth. All right, let's go back to the description. Where are we? Okay. Yes, just to reinforce this, we have a vector of npat possible sample sizes, SAM size vec that can be selected from randomly, which is the second input to the following function. All right, so here's my SAM size vector. That's this vector here, vector of possible sample sizes. Okay, I kind of got my ahead of myself a little bit here. That's the process that's described in the following paragraph to create simulated samples. So write a function that takes as inputs a matrix of simulated patient counts and a vector of possible sample size. When I say matrix here, I mean an array. So sometimes I say matrix, but whenever I say matrix, I really mean an array. Okay, so this is also an array. Okay. So simulated patient counts and a vector of possible sample size. The output would be an array called SimSam matrix that's the same size as the array of simulated patient counts. So that's this array down here. This is what would be called SimSam matrix. So maybe I'll just call that here. This is SimSam. Okay. And then this one is SimPatMix. And this one here, the gene, this was, what did I call this one? I called it, where is the name? ReadMix. This is ReadMix here. All right. So here, so how would you simulate these numbers? I'm going to have a simulation again for 0, 1, and 2, because those are my genes. Okay. All right, so how am I going to generate these numbers? To generate samples, turn the row of the simulated patient count array into a probability vector by normalizing so that the sum is 1. And use these probabilities to draw a sample with replacement of size n read from the set of numbers 0 up to ng minus 1. So what's going on here? I want to generate this many reads to fill these columns, but I want to do it according to these proportions, right? So in order to do it according to these proportions, I have to take this proportion here. So this is going to be equal to this value divided by the sum of these values here, okay, because that's going to give me my proportion. All right, but I want to make sure that this carries. So here I want to use the, well, all right, let me do the scan. Here, let me make the sum. So I'll do sum. So equals sum. Right. And we'll copy that down. Okay. All right, so here it's going to be proportion. So this will be equal to this value divided by this sum. Okay. And in order to make this carry to my other cells, I want to always take the sum from column L. Okay. And I can fill down, and I can fill across, and then this should sum to 1. Okay. All right, so these are now represent the proportions of these counts. So I'm going to choose randomly 5,003 numbers according to these proportions. So when you generate A prime, it's not going to look like this. When you generate A prime first, you're going to generate a sequence of 5,003 numbers, and those 5,003 numbers will be among 0, 1, and 2. So you first will generate something like 0, 0, 1, 2, 2, 0, 2, etc. So you'll generate 5,003 of those numbers. Okay. From that sequence of 5,003 numbers, you're going to extract the counts of each gene. Now, it will probably be, you have to try to use as few loops as possible and try to do this as efficiently as possible. Otherwise, your code will be very, very slow. So the final result will be a set of counts here such that the counts sum to 5,003. And the proportions here will be roughly the same as these proportions. And you do that for every single patient. Okay. Now, by the way, when you're testing your code, I would recommend testing with a relatively small set of patients and a relatively small set of genes. You don't have to use the entire read mix at the beginning to do your testing. You can take a sub-selection during your testing, and then finally, once you've made sure that your code is correct, you can use the entire data matrix. Now, you should test your functions one by one, and there are various things you can test to make sure it's correct, make sure the sums are correct, make sure the values are correct, etc. So please test with a small data matrix, and then you can apply it to a larger data matrix. All right. So I think I finished describing this. You take a row of a simulated patient count into a probability vector by normalizing so the sum is 1, and use these probabilities to draw a sample with replacement of size and read from this set of numbers. Then convert your samples back to counts. Okay, so that's what I was saying here. You'll create this vector here, and you'll convert that back into counts. So this will give you a vector of length n gene, which will be a row of your matrix, SimSam matrix. So you see that the number of entries in this row is the same as the number of genes. All right? All right. So these will be your observations. Again, these are your observations that are trying to be used to predict these. Okay. So you have two matrices, SimPatMix and SimSamMix. The latter, in some respects, should be an estimate for the former. Okay. So the latter is observed and the former is ground truth. So we're interested in the relative recounts for corresponding genes for two different patients. All right. So we want to compare across patients. We want to determine if patient P has an elevated gene count of gene G. We would compare SimPatMix PG to the mean value of SimPatMix over all patients. Okay. But we don't have SimPatMix, we have SimSam matrix. So we'll try to use SimSam matrix to get this ratio. Okay. Like I said, if I take SimPat matrix for patient P and gene G, so if that's bigger than the mean value over all patients for that gene, then that patient has an elevated value for gene G. That patient P has an elevated value for gene G. So his relative level, relative to the mean, determines whether his gene level is elevated or not. Okay. And again, we would like to do that with SimPatMix, but our observations are SimSamMix. So can we use SimSamMix instead of SimPatMix to get this ratio? Okay. So compute the means of each column of SimPatMix and SimSam matrix. You're going to need those means. So you're going you'll get SimPat call means and SimSam call means respectively. Okay. So that will be down here. Right? So here I would take the mean. This would be the SimPat call means. So that would be taking the column means of these columns here. So in Excel, it's average. They don't have mean. So it would be average of this. Okay. And do this across. And you would do the same for these here once you fill these in. Right. So I don't know, just fill this in manually through 35. And, okay. So here you just take equals average of this. Right. So you're going to compare this proportion with this mean. So this would be SimSam mean. All right. Or let's. I guess it's not SimSam mean. It's SimSam call means. Sorry, SimSam call means. All right. So all right. Now divide every row of SimPatMix by SimPat call means to obtain pat-rat mix. Okay, so what do I mean by pat-rat mix? Pat-rat mix is the ratio of each gene for each patient to the mean of that gene over all patients. Okay? So to create pat-rat mix, you take this column, divide by this, and take this, you take this row, divide by this, also take this row and divide by the same. Divide every row by this column mean. And there's a way to do that efficiently in Python. So try to do it as efficiently as possible. You should be able to do it without a loop. Every time you do a loop, you're losing time. So you're going to create those ratios. So you're going to do so you can do pat-rat mix and sim-rat mix. Okay, you're going to do the same thing with this one also. And that will give you your ratios. Okay, so the entries of sim-rat mix should approximate the entries of pat-rat mix. Okay, we're using our observations to approximate ground truth. So define error matrix as the difference between the two matrices. Okay, so that's going to give me my error matrix. For each column of error matrix, take the mean of the absolute value, and the mean and the square root of the mean square. Right? These are three statistics that tell me about the distribution of the error for each gene. Right? So let me show you down here in my example sheet. So if I have my error matrix down here, let me just fill this in with numbers. Now the errors can be positive or negative. So let's say this is 0.1 and this is minus 0.2, something like that. Okay. Right. So the mean, of course, would be the average. Now to get the mean absolute value, so this would be the mean. The mean absolute error would be the mean of 0.1 and plus 0.2. I can't do that conveniently in Excel, but that would be like 0.15 would be the mean of the average. So this would be the mean average error. Okay. And the mean squared error would be, there's a way to do this in Excel. You do sum square, right? Would be the sum of these squares. However, I want to take the square root of that. So that would be the square root of the sum square. Right? And this would be the root mean squared error. Okay? So this gives me statistics for the errors for each gene. All right? Hopefully, these errors are relatively small. The errors are kind of in percent because I'm talking about the errors in proportions. So the error is sort of in percent. So it's like 22% error. Right. So let's see. Right. So the first statistic will give me bias. Second will give me the mean absolute errors. Third will give me the root mean squared error. Okay. And each of these vectors will have length ng because I'll be computing one of these. I'll be computing this for every single column. Okay. All right. So we want to see how these errors depend on various factors related to the genes. Okay. All right. The different genes will have different errors. Some genes may be more accurate than others. So I want you to do the following scatter plots. So you'll have to decide for each plot whether or not to use log scale on the x or y axes. You can tell by how the points are spread out. Right? If you do a plot on regular axes and you're getting something like this, let's see if I can do this. Okay. Is it working? All right, I guess it's not working well. If you're getting data where you get something like you have a lot of points way down here, and most of your points are down here, and then you have a few points out here. Right. That's an indication that you want to use a log-log scale. Or it may be that happens on one axis and not on the other. So maybe these are clustered here, and then these spread out like this. So in this case, you'd put a log scale on the x-axis, but not on the y-axis. Right? So plot your data so that it looks well distributed. You want data that looks that you can see the shape of. So it'd be a nice cloud, and you can see the trends and tendencies. All right. So these are the different graphs that you'll be doing. You do these three statistics on one axis versus the column means of SimPatMex on the other. Okay. And the column means are the mean expression levels. Right? We already have those column means here when you have SimCat column means. Okay. All right. Now you can also do this, also plot it versus the column standard deviations. Right? And you can also do it versus the column relative standard deviations. Now, if you have problems with division by zero, then just remove the columns that divide by zero. It's possible that some of them have a mean of zero, but in that case, you can just remove that column. All right, now, two more plots, these same statistics versus the logarithm, okay, and the mean of the logarithm and the mean of the standard deviation. So the logarithm kind of equalizes out the data to put it on a more compressed, more realistic scale. Not more realistic scale, but a more even scale. All right, so you got all these graphs. Now, hopefully you'll only get 5 graphs. It would be nice to plot these 3 on the same graph. They may not show up very well, but try to represent the data as best you can. All right, and then just one more thing. So this should be easy because you already have the code. Let's see if we can do something to reduce the errors. Now, I asked you way up here to sort the columns so that in order of increasing standard deviation. And the idea is that the genes that are highly variable mess up the other genes. So we can see if we can remove those genes and do the same analysis and see if we get less error. Okay. So remove the final 1,000 columns of read matrix and repeat the entire analysis. So you just copy your code and do the entire code with a different read matrix and compare your results. Then write up what you've learned about this estimation problem. Can you think of a way to reduce the errors? So the question is this an accurate method for using the I have SimRatMex to estimate PatRat matrix? Are we getting fairly good errors? And how do these errors depend on the genes themselves? And then does this process here reduce the error by removing the genes that vary a lot? Okay, that's it.